into our lab. Just want to give a brief introduction to our lab. So the Nuzhdin Lab at USC actually has its origins in fly biology. So we have sort of a long history of fly genetics and population genetics. And more recently, we've moved to omics, including genomics, transcriptomics, and hopefully more methods soon to investigate domestication events in like plants and other like domesticated species, but also more recently we're turning that focus into kelp. And so this is our lab. This is my PI, Sergey, and we have some photoshopped people in here. And then more recently, this is what our lab has tended to look like, but we're hoping to get back into USC and working soon. So the project that our lab has with Giant Kelp is actually funded through a facet of the US Department of Energy. And it's kind of surprising. I thought that they at first were working from an ecological standpoint, but this program through ARPA-E called Mariner is actually looking to develop basically parts of the US and tools in the US in order to produce more marine biomass. And in this instance, the application that this program is really interested in is biomass for biofuel production. But there's like a bunch of other applications as well that these projects are looking at, including as a feedstock for livestock and cattle, or also as a supplement in human diet and things like that. So I'm going to start with, if you've come to probably any other of these kelp magic talks, you probably know this, but brown macroalgae or kelp are extremely ecologically significant. They are found globally, and they are actually pretty distinct from plants. So green algae, I believe, are actually from the plant strain of evolution, but brown algae have actually evolved completely separately to happen to look and function a lot like plants, which is, I think, pretty cool. And they are also habitat forming, mostly. Like a lot of Saccharina and Macrocystis, Neurocystis, like bull kelp, are form these large structures in the water, like you can see here, that provide habitat for hundreds of species of fish and wildlife in the ocean. So the part of seaweed growth that I want to talk about today is actually the industry applications of seaweeds, which I did not realize there were so many until I joined this lab. And it's actually really important in the global economic sphere in a way that I was unaware of. So obviously, one that you might already be familiar with is seaweed salad, which is often a species of Undaria that's found in Japan, but has been cultivated in other places. But red algae is actually used in a lot of applications as well. So it makes this like gelatinous sort of substance that's used. If you are in a lab setting ever, you've probably used agar to form DNA electrophoresis gels, but it's also used as a gelatinous substance in baking and in cosmetics. So the big application that I want to talk about today, of course, is turning huge amounts of seaweed biomass into biofuel. So why not agriculture is kind of the first question that we can come at this idea with. So agriculture has been used as a biofuel resource in the production of ethanol, bioethanol from sorghum and corn and things like that. But climate change is having a huge threat to agricultural yields. And agriculture also requires far, far more inputs of energy and nutrients than aquaculture. So aquaculture, for instance, would not compete for fresh land, fresh water, or for arable land and fresh water, and fertilizers and nutrients and things like that. The other reason that there's a lot of interest recently in the use of biofuels in the global fuel sphere is that the demand for fuels that are not fossil-derived fuels is increasing, and the production of biofuel, such as ethanol and biodiesel, has actually increased significantly over the last decade. Brown macroalgae is in a lot of projections been targeted as sort of the next evolution of biofuel. So agriculture and especially crops and including corn, sugarcane, and also like larger plants such as like trees have been proposed to be used as biofuel resources. And the sweet spot that we're kind of hitting right here is the third and fourth generation of biofuel production, in which the resource has been proposed to be marine for kind of a lot of the reasons that we've mentioned, or that I mentioned previously. So lower inputs, doesn't compete with agriculture to produce food, and it doesn't take as many resources. And it's also extremely scalable. There is a lot more ocean than there is land on this planet. And a lot of our research is going towards doing aquaculture in a way that is able to take advantage of basically the open ocean and the amount of resource that we have that's available only with aquaculture. And the aquaculture industry is actually kicking off in the US. This is kind of new and recent because a lot of aquaculture has been focused in other parts of the world, especially Asia. But there's a few industry partners that we have that I've listed here that are scattered all over the US, and they're taking whatever kelp species are native to kind of the places that they're nearby and setting up farms in order to harvest more like volume of these kelp species for, I believe Blue Evolution's involved in like food production. Green Wave is involved in integrating to trap waste from fish aquaculture farms. So the sort of major difference that I want to highlight between the culturing of crops nowadays and the on land in agriculture and the culturing of kelp in the open ocean through aquaculture is that kelp is wild. There's no necessarily like domesticated species of kelp. There are certain crosses that have been formed in like Asia and have been used for breeding, but on the whole, it's many species of kelp are not domesticated and are not well understood enough to domesticate them efficiently. So something I want to highlight in terms of our approach toward domesticating kelp for this project is that corn, for example, that I've used here is actually derived from this ancestor plant called teosinte that looked very, very different from corn. And this time scale of domestication by phenotypic selection of humans over and over, selecting for taller stalks, more kernels on the corn, is on the order of thousands of years, if not hundreds of thousands of years. And so this is basically super limited way of domesticating and breeding a species. in order to turn it into a crop. And what this is susceptible to is that if you have this like super good advantageous allele in the genome that you want to kind of keep around in your domesticated crop, it's really hard to do a crossing scheme that can get rid of any linked, maybe deleterious mutations nearby to this allele that you're selecting for. In contrast, we're proposing to use genomic domestication in order to more efficiently domesticate kelp and to kind of end up with a better product. So in the event of a genomic domestication, you can basically intelligently select for an allele of interest, and you can use either genomes that you know do not contain some linked allele that you don't want, or you can use genetic modification, whatever permitting sort of issue is in your way, and introgress certain advantageous alleles that you would like. And this can be done on the order of years once you've developed the genetic tools for a certain species. So the species that we're super interested in is Macrocystis pyrifera, or giant kelp. So this is the kelp forest forming kelp that you'll see off the coast of California. And we actually have partnered, this is a paper from one of our lead PIs on this project. And basically we found that the area right off of Los Angeles and basically the Southern California Bight contains the bulk of genetic diversity in giant kelp, though the range extends far beyond north and south of the Southern California Bight. This is actually proposed to be the origin of the giant kelp species because the concentration of genetic diversity is so high in this area. And so with this valuable resource, basically this kind of pot of gold of all these alleles to choose from and to sort of investigate and find the best alleles for domesticating giant kelp for biofuel applications, we have proposed to do genomic domestication basically through this schema where we have collected basically a seed bank. So this is, these gametophytes are baby kelp, and we're going to go more into the life cycle of kelp later and kind of understand how this flows. But basically the design is we've captured from this pot of genetic diversity in the Southern California Bight a bunch of like seedling gametophytes that you can use for breeding. And so the test farm that we've created 2 years running now is from crosses of one male with 500 females. And we do that with 5 times replication, and we outplant that. And basically we're trying to see how kelp is growing in a seed farm, like from with samples from all of these different genetic backgrounds, and see which genetic background is going to give us sort of the best performer in this sort of fixed environment that we're growing them in. And so this is kind of the overall view of how our project and is similar to the way that a lot of other of these mariner projects that are using giant kelp and other species of kelp to create increased amounts of biofuel. This is kind of the overall schema for this genomic style of domestication. And something I want to highlight is what is actually the desired phenotype of domesticated kelp. And something that's really interesting to me is that kelp is attractive because it contains sugars, but also attractive because it contains lipids for different reasons, depending on the method that you would like to create biofuel from this species. And so I think it's really interesting to look at how different genotypes that we have in our collection are accumulating biofuel relevant molecules like lipids and sugars. And we can use some of the tools in our omics suite, like Metabolomics, in order to get at these questions. Something else that's basically one of our biggest goals in the farm is to increase growth and the accumulation of overall biomass, which we can use more simple models of basically, we can do genome-wide association studies in order to find which alleles are contributing to increased growth in biomass. And one of the sort of auxiliary, but also very important sort of steps that we're taking towards domestication is actually sterilizing our outplanted genotypes. So we haven't been able to do this necessarily yet in the farms that we've made, but we're working towards identifying mutations that exist in our sort of pot of genetic diversity that we have. And we're looking to isolate some mutations that when combined together to make a kelp from sperm and egg might create a sterile outplant. And this is more to mitigate invasiveness and things to do with, you know, if you actually want to have a kelp farm, you have to worry a lot about permitting it and making sure that it's safe and not disturbing natural wildlife or creating invasive species. So this is an overview of our lab's analyses toward domesticating Macrocystis pyrifera or giant kelp with genomic tools. So we've done DNA sequencing, some RNA sequencing, and we're basically combining a lot of our analyses in order to domesticate kelp by basically finding the genetic basis for these desired phenotypes that I just discussed, and also trying to find mutations that could induce sterility in our genotypes of interest. So the first topic I want to go through is metabolomics and how we can apply metabolomics, or the study of small molecules present in an organism. We can basically combine some of this information about what lipids and sugars and other important molecules are present in different genotypes, along with our knowledge of the genetic background of those genotypes and what genes they're actually expressing through RNA-seq, and identify how we can create more of these biofuel relevant metabolites that we want for biofuel production in our domesticates of giant kelp. And so, yeah, here, and also towards high biomass yield. That's actually something we can do with metabolomics as well. So as a primer, in case anybody is not familiar, metabolomics is the study of both primary and secondary metabolites. So, for example, primary metabolites would be involved in this central metabolism, like TCA cycle, glucose, digestion, production of ATP, and synthesis of things like polyketides and terpenes and alkaloids would be considered secondary metabolism. So this whole sort of L shape is all central metabolism and primary metabolites, and also these secondary metabolites. And so what these can tell us, basically by sampling from, for instance, a genotype on the farm, is kind of give us a snapshot of what all metabolites are present in a certain genotype. So maybe you're interested in this genotype and you want to find out, what's responsible for it has a high amount of lipids or something that you're interested in for biofuel. And you can go back and see which actual mechanisms are being used to produce these metabolites of interest based on what metabolites are present. So an example of this is actually really easy to see from agriculture, which is definitely farther along in the application of omics for sort of improvement of crops. But this paper that I'm referencing does something really cool where they basically take this like metabolomic profile, like this snapshot of a bunch of inbred lines in a plant, in maize, actually, excuse me. This is in corn. And so using this metabolomic profile and the associated phenotype, they were able to basically do machine learning in order to understand what metabolites are important for stress tolerance in maize. And so the stress here is salt tolerance. And so they added salt as a treatment, and they separated these inbred lines into high salt tolerant and low salt, or excuse me, high salt tolerant and low salt tolerant lines. And they were able to actually isolate 37 metabolites that are super important for tolerance in this species of corn. And so doing things like this can be really important for designing breeding in the future if you know that you're going to be dealing with higher heat, for instance, with climate change, or maybe with storms or with high salt content, as in this instance. Metabolomics can be really important for kind of isolating what exactly is going on underneath the hood. Like, why are we seeing this effect in certain genotypes and not in others? So in turning back to aquaculture and kind of taking what we learned from agriculture, can we basically look at the metabolites present and the pathways present in giant kelp in order to select for more biofuel relevant metabolites? And this is not necessarily an easy question. The reason for that is there's, as you can see from this diagram on the left, about 10 different ways just from this paper alone that have been identified to produce biofuel from kelp. And this is pretty general, not just for giant kelp. This is for just like marine kelp biomass. But each of these procedures is going to rely on either higher sugar concentration or the concentration of specific sugars for digestion by bacteria or yeast. And then in bio-oil and biodiesel, and especially to produce chemicals that aren't even necessarily just fuels, but also plastics, focusing on like lipids and cholesterols and things like that, and that increasing that concentration is going to be really important. So basically, we want to use metabolomics to investigate what metabolites are we seeing present in the profiles of our genotypes on the farm, which are created from crosses from those populations that we were looking at earlier. And so what genetic backgrounds are maybe producing more lipids or more sugars, and can we basically figure out if there's a way to supplement that pathway with maybe something that we add in a hatchery stage before these are outplanted on the farm? Is there some mutation that's super important in maybe like increasing the efficiency of some pathway? Things like that we can look for. And something else that I mentioned is that biomass growth can actually be the question of like what's increasing biomass growth can be sort of tackled through metabolomics. So this is a example from diatoms, which are extremely distantly related to brown algae, but are close enough sort of evolutionary relative to be relevant in this case. So these are really tiny organisms. I don't actually know if they're single-celled or not, but they basically have been investigated to see if they can do genome engineering, so that you can actually take hold of all the metabolites that are in the cell and plug that into a model of how basically how carbons and how nitrogens and how energy is shifting through this individual. And so you can use genomes and annotated genomes in order to understand, all of the proteins that are present that are acting on these metabolites, and sort of pushing them through these different pathways. And so you can actually use this to model, which increase in which metabolites would create the increase of biomass, or create the largest increase in biomass. And so you can use these basically genome scale models, and something, if you're interested in this at all, this has been modeled a lot more in things like yeast, and it's called flux balance analysis. And this is another advantage of using metabolomics to sort of get at the question of how to sort of efficiently domesticate giant kelp. So that's going to kind of conclude me talking about the domestication for like biofuel phenotypes portion. I kind of want to move into the topic that I hinted at earlier, which is sterility of these species. So what's really important to consider when we're talking about having these offshore farms or these open ocean farms of different kelp species for biofuel production, or even for other applications for industry. It's really important to consider kind of what we've learned from the history of aquaculture, especially when people have tried to culture species that were non-native in different areas. So a very infamous example is Undaria pinnatifina, which I mentioned earlier, is often used to make seaweed salad. And basically, this edible brown kelp was super popular, and people really wanted to grow it in other places. And then I think it has also been moved around a lot by ballast water. But this species is now super invasive on French, Spanish, and UK coastlines. It was listed in like the top 100 most invasive species list one time. And of all known non-indigenous seaweeds globally, kelp account for 25% of non-indigenous species in marine environment, which is pretty significant to be a fourth of all the types of species that are non-native. And so when we go to permitting agencies to try to put out these farms, or when industry partners try to put out these farms, permitting the growth of these improved varieties of kelp is basically a huge ecological concern. And in being sensitive to that, and in trying to kind of create this more sustainable way of producing biofuel that's non-invasive, we've worked with some collaborators who are on another Mariner project to domesticate kelp. So the project that we're partnering with is hoping to domesticate sugar kelp, Saccharina latissima, on the East Coast. And these species are fairly closely related. They're both brown macroalgae species. And in both of their genomes and transcriptomes, we're looking for genes that are involved in reproduction, and we're looking for basically mutations that can help us sterilize these species when the mutations are combined in our breeding program. So to kind of clarify how you can sterilize something that you're outplanting without basically killing your whole breeding program, I wanted to go through the process that we use, at least for our project, and I believe for the Saccharina latissima project as well. We basically take zoospores released from a sporophyte. So a sporophyte of kelp is basically just the macroscopic large structure that you see in the ocean. And this is the diploid stage of it. So it has 2 copies of the genome. And sporophytes will produce specialized tissues that will release zoospores. And instead of just releasing sperm and egg that mate, they actually have this intermediary step where they produce gametophytes. And this is actually the only instance in which sex is expressed in a lot of brown algae that follow this life history. So it's a UV sex determination system. If you have one type of chromosome, you'll be female. If you have the other type of chromosome, you'll be male. And these multicellular microscopic structures are actually what produce sperm and egg and fertilize a zygote to become a sporophyte. And something that's really important here that I've kind of highlighted in red is that red light can actually arrest the development of these male and female gametophytes when they are grown in lab. And so you can keep these sort of in a pre-hatchery stage, like before you cross them to create a zygote. You can just keep these immortalized male and female lines of kelp gametophytes. And this is how both giant kelp and sugar kelp actually have this life history style. And what this means is that we can take genotypes and kind of keep them endlessly. So if we find the best combination of a few, handful of genotypes that make the fastest growing kelp or the highest sugar content kelp or something like that, we're able to immortalize those in our lab. And so in order to sterilize whatever we outplant from these, basically the way that we tend to think about these gametophytes is as seeds, right? So if you have a bank of seeds, you can basically outplant the seeds and maybe make an apple tree that doesn't have any seeds in the apples themselves. So they're effectively sterile. And so what we've proposed to do in order to sterilize these kelp species is to make them sporeless. So that's the tagline is sporeless kelp. So you can use life cycle manipulation either with just informed crosses, which is probably going to be a lot easier to permit than using CRISPR or something like that. But you can induce a mutation that you can induce mutations in these gametophytes that when crossed, the sporophyte that they go to produce will be diploid and will be homozygous for whatever mutation you choose, or it could be a combination of a few mutations, and these will prohibit the sporophyte from undergoing meiosis and producing zoospores. So that's kind of why we have it called sporeless kelp, because these zoospores shouldn't be produced when the macroscopic stage is outplanted in the open ocean. And there's pretty good conservation across a lot of brown macroalgae species of at least what's going on in meiosis and in zygote formation. And so we can sort of look at genes that are involved in reproduction by looking at the specialized tissues that produce these zoospores and contrasting that tissue with non-reproductive tissue to try to see beyond what's been identified in these simpler like green algae. I think this is a green algae model. We can look at what other genes have been conserved or what other genes we can find that maybe are new that are involved in reproduction in brown macroalgae specifically. So the experimental design that we've used for this sampling, my colleagues are working on basically an analogous version of this experiment in giant kelp, but for now I'm going to be talking about sugar kelp or Saccharina latissima. And so Saccharina latissima has this long blade that it develops, and then this holdfast is connected to the seafloor. And so all this tissue that is along the blade is non-reproductive and is typically just referred to as like blade tissue or sometimes blade ends if you're like clipping from here. And then the source tissue will develop in patches basically along the blade. And this is the reproductive tissue and it contains these zoospores. So this is basically the tissues and the cells in which meiosis is happening. And so what we're hoping to do is compare the RNA-seq results of looking at this source tissue and this blade tissue in sugar kelp and finding basically what genes are more expressed in source tissues. So which genes are we finding that are actually involved in reproduction? And so these putative reproductive genes of interest will be highly significantly more expressed in source tissue. And so this is a volcano plot, which is a way of looking at differential expression of genes in different treatments or tissues. And so here we're going to be really interested in this top right panel because it's going to show basically the IDs of whatever genes are involved in reproduction putatively. And so there's certainly sure to be a lot of other genes that are, involved in procedures or that are involved in pathways that are not necessarily reproduction oriented. But we're hoping that in this pool of genes that we've identified from this analysis, we'll find reproductive genes. And then the step that we're going to take after doing a functional analysis of what popped out of this differential expression analysis, once we kind of find out what these genes are and what they're doing, we're going to apply some of the mutation annotation work that I've been doing. So I've constructed a pipeline in order to identify the effects of different types of SNPs. And basically, this is preliminary at the moment, if only because we only recently got access to the brand new Saccharina latissima genome. And so this has all been done by aligning to transcriptome. But in the future, this analysis is going to be more robust in order to identify mutations across the entire genome. And so just to validate this pipeline, we see that it's correctly identifying that most SNPs are going to be in the third position of a codon. And this just makes sense because there's a wobble position in when translation is happening with RNA codons. And we tend to see more mutations in this third position because it's a little more forgiving and typically doesn't change the protein that's produced. Now, in the evaluation of all the SNPs that were called just on the transcriptome, almost 20% of them were coding. This is pretty high, but we're also calling it mostly on expressed genes. And so genes that were not in intronic or UTR regions would be in coding regions. So that makes sense. And then we also can start to look at what I would call some of the juicy information here on the bottom. So this is where it starts to be cool. We see a lot of non-synonymous SNPs. So the effect of the SNP is that it changes the amino acid that's going to be produced in the protein. And that doesn't always have a hugely deleterious effect, but it can in certain cases, depending on if that amino acid is directly involved in the binding site or something like that for the protein. And the other super interesting little tiny pink bar here is that we found just about 1,600 nonsense mutations. And this is from using the whole genome sequencing of 50 Saccharina individuals. And so that's a good amount of nonsense mutations. And the reason that those are exciting is because they introduce an early stop codon in the sequence of the protein in DNA. And so when that protein is encoded, it's often artificially short and can sometimes knock out the expression of the protein overall, or at least the function of the protein in the cell. And so the other thing that I did is tried to characterize what some of these non-synonymous SNPs mutations were. And so you can kind of characterize, you don't know exactly how the SNP is going to affect the function of the protein, but you can categorize these SNP changes into radical and conservative changes. So a radical change would be like an amino acid that is polar and changing it to non-polar, or an amino acid that is hydrophobic, and then you switch it out for a hydrophilic one. So those are usually the categories that we use to determine radical and conservative changes. And we see a lot of radical amino acid changes that we can investigate. And the other thing too is the previous analysis that I mentioned will help us figure out which of these mutations is in a putatively reproductive gene. And that's sort of going to guide our analysis. And the last thing I wanted to show is that we have a pretty wide distribution of these nonsense mutations across all the genes that were called. And so I'm pretty hopeful that we'll find a mutation in a reproductive involved gene. And hopefully we can, let me go back to the graphic that I was referencing. Hopefully we can make the dream of sporeless kelp a reality by tackling sort of figuring out what the effect of these mutations are that we've identified. So that is all that I have for today. Thank you guys so much for listening. I want to thank my collaborator, Scott Lindell, especially for working with me on the Saccharina latissima work and providing a lot of the data. And then Philippe Alberto is our PI for the Giant Kelp ARPA-E project. And this is my contact if you'd like to reach out. And thanks to everyone in the lab. I'll take any questions if you guys have any.